talking about mitochondria. The main function of mitochondria is aerobic respiration along with formation of ATP. We know that they have their own protein synthesis machinery. They are semi-autonomous organelles. Now when we come to the structure, we clearly know it's a double membrane organelle. This is the outer membrane and the inner membrane is folded to form cristae. These are the foldings of the inner membrane and these are known as cristae. It has perimitochondrial place. This is the mitochondrial space. Then it has small circular bodies which are known as ribosomes in it. Then it has oxysomes which are attached onto the cristae. These oxysomes are present everywhere. This inner one is the matrix. These ones are oxysomes. And here small circular DNA is observed along with the ribosomes. So this is the outer membrane and this is the inner one. Outer membrane, inner membrane. In between is present the perimitochondrial space. Ribosomes are there, cristae, oxysomes, matrix. This is all about mitochondria. Here we say it is a double membrane structure and accounts for aerobic respiration, formation of ATP as well as storage of energy takes place here in the mitochondria because of oxysomes. And oxysomes, they are the ones which are attached to the membrane like this. This is F1 subunit. This is F0 subunit. And formation of ATP takes place here in this F0 F1 subunit with the help of enzyme ATPase. These oxysomes, which are located on inner side of mitochondrial membrane, help in the synthesis as well as storage of ATP. Then are plastids. Double membrane bound organelles of different shapes but mainly found in plant cells, euglenoids, responsible for pigments because they contain pigments as well as the storage products. There are three types of plastids which are commonly found. These are then chromoplast, leucoplast and chloroplast. It depends on their color and function what kind of plastid it would be. Now when we talk about the first one, leucoplast, we say these are colorless, usually found in storage organs. Storage organs are tubers, fruit, rhizome, etc. They are mostly not exposed to sunlight. Then we talk about their main function. Main function is to store starch. If it stores starch, we call it amyloplast. If it stores proteins, we call it eleuroplast. If it stores fat, we call it eleoplast. Okay, moving to the second one. Chromoplast. Chromo stands for color. So these are the colored plastids. These ones were colorless. These are colored plastids. And they range from yellow, orange to red pigments. So basically they have carotene and xanthophylls. Where are these found? Found in petals of flowers, and skin of fruits, right, which is varying from red to yellow to orange in color. Then they attract agents for pollination, and dispersal of fruits as well as seeds. So basically what is their main purpose? They are helpful in pollination, in fertilization, as they attract pollinators, and pollinators help in the dispersal as well as in pollination of food. Then is pollination and dispersal, that is mainly you have to keep in mind. The third one is chloroplast. Chloroplast, the green plastids, mainly known as only green plastids, and they contain some amount of, mainly they have chlorophyll, this which is responsible for the green color, chlorophyll A, B, C, D, and then they have, along with the chlorophyll, they have carotene, then xanthophyll, and this is present in the thylakoid membrane. This you have to remember. Thylakoid membrane has this chloroplast, right? And what the matrix does? Enzymes for the biosynthetic phases of photosynthesis. Even they have this chloroplast that we basically talk about. This chloroplast, it has its own DNA, circular DNA, 70S ribosomes. These are also semi-autonomous. We are going to draw the picture of chloroplast. This is a double membrane layered organelle. See, if I open up the outer envelope, this is how it will look like. If we open the cover, and inside there are located thylakoids, stacks which are present onto each other and are interconnected. So this is the outer membrane, and the inner membrane, the one which is here, it is mainly forming the thylakoid also. Okay, inner membrane. Then granum, and basically call it thylakoid. Then it is the stroma lamellae, stroma lamellae, and this region is stroma, which contain ribosomes, then it has circular DNA also, and we see that the matrix which is present here, matrix is responsible for the biosynthetic phase of photosynthesis, as it contains enzymes, and it also has its own protein synthesis machinery that we know. It is all due to the single circular DNA. S ribosomes and RNAs. That's why we call it semi-autonomous organelle. Then moving to what are ribosomes. As we know about ribosomes, they were first observed under electron microscope, and they are made up of, ribosomes are made up of ribonucleic acid and proteins. They do not have any boundary, no boundary membrane, means no enclosure. These are of two types, ATS and 70S. If we say it is ATS, it is composed of two subunits which are of 60S as well as 40S. If we say it is this 70S, it is composed of 50S and 30S, and this is very clear to you that 70S is present in prokaryotes and ATS is in eukaryotes. And the main function associated with ribosomes is protein synthesis, right? Now we are moving towards the cytoskeletal structure. What do we mean by cytoskeletal structure? Cytoskeletal structure is an elaborated network of filamentous proteinaceous structures. Basically, microtubules, microfilaments in the cytoplasm. Cytoskeleton structures. It is a proteinaceous structure which is formed of, proteinaceous is microtubules, microfilaments, right, which are formed in the cytoplasm. Now, cytoskeleton, it performs the following functions. What? It provides mechanical support. If we call it cytoskeleton, obviously mechanical support. Number 2, maintains shape. Number 3, regulates the distribution and orientation of cell organelles. Number 4, helps in cell movements. This is what it does. Okay, the first one that I'm taking here is cilia and flagella. It is an hair-like outgrowth of the cell membrane, basically small and work like oars, causing the cell movement and for the surrounding fluid inside and outside of the cell. This is how the cilia and flagella work. Cilia is basically smaller and flagella is longer. Here we have an array of 9 microtubules which are present on the outer side, and these 9 microtubules are 1 complete and 1 half, second, third, fourth, fifth, sixth, seventh, eighth, ninth. Here 1 complete ring and 1 half ring. These are present. All these peripheral microtubules, these are attached to each other with the help of a connector. See. Internally, they are connected to the central microtubules, which are connected to each other with an interdoublet bridge. This central sheath is supporting all the peripheral 9 microtubules with the radial spokes. These are the radial spokes with which all the peripheral microtubules are attached or are connected to the central sheath. They are connected. Right? And there are small spokes which are connecting all the peripheral microtubules to each other. So these ones, let's make it a circular in appearance. These ones, the outer ones, black ones, these are the peripheral microtubules. Peripheral microtubules. These are 9 in number. Then this one is the central sheath. Then is interdoublet bridge, which is connecting these two together. Interdoublet bridge. Then these are the central microtubules. These are 2 in number. Central microtubules. Then there are the radial spokes which act like connectors. These ones. We call them radial spokes. And the outer one is always the plasma membrane. Now, the structure says that cilia, it is relatively smaller work like ore, this causing the cell movement. Obviously, flagella are responsible for cellular movement, but these are relatively longer. Okay, these may help in movement of surrounding fluid or substances inside the organs as well, but this is restrictively responsible for cell movement. Since it has 9 and 2 internal or central microtubules, so we call it 9+2 arrangement or 9+2 array. Okay, this is called 9+2 array of the cilia and flagella structure. Moving to centrosome structure or a centriole structure. It is also a cytoskeletal structure, we call it. Centrosome. And centrioles. Now it consists of pair of centrioles at the right angle to each other, surrounded by a pericentriolar cytoplasm. See, basically in a cell during the time of division, these appear in this manner. Okay, a centrosome consists of a pair of centrioles at right angles to each other, surrounded by an amorphous pericentriolar cytoplasm called centrosphere or kinoplasm. They form the basal body of cilia and flagella, and from here the entire spindle apparatus starts at the time of division. We are talking about this centrosome and centriole in structure. Okay, and here we say it's the central hub to which 9 peripheral microtubules arise. It has a complete ring and 2 half rings. A complete ring and 2 half rings. And we mark them as A, B, C from the center to the outer side. This is A, B, C. Clear? C is connected to A. In every side, the C is connected to A. It gives a star appearance. This is the central hub. This is the central hub. It's the wheel of a cycle, or you can say the star shape, right? And all this is made up of protein, tubulin, and these are the radial spokes which connect all the microtubules. Now these microtubules is 1 complete ring and 2 split halves which are connected to each other. 1, 2, 3 in this way. This is A, B and C, and same way the another A, B and C. A, B and C. So here the C is connected to A of each microtubule like this, forming an arrangement of a star appearance. Since there is a central hub, there is no microtubule which is there in the center. This arrangement is known as 9+0 array. What do we call it? 9+0 array. Alright, moving to nucleus. nucleus. Now, it is mainly responsible because we say that nucleus is something which is the brain of the cell. It contains all the information, necessary information which is required for the cell to work. And various scientists came forward. Robert Brown described it nucleus. Fleming gave the name chromatin to the material in the nucleus, which is basically on the stained, right? We know this nucleus, which is here, it has an outer as well as an inner membrane. So we draw it like this. That it has an outer as well as an inner membrane, and it has extended endoplasmic reticulum along with that we know. Ribosomes attached onto it. It has nucleolus, nucleoplasm, nuclear membrane, nuclear pore, nucleoplasm, and the chromatin network inside. This is the chromatin network. At the resting stage, this chromatin network remains as it is, but at the dividing stage, it gets condensed to form chromosomes. Right? Each eukaryotic nucleus has a double membrane boundary, space between them, which is known as perinuclear space. Nuclear membrane forms a barrier. Outer membrane is more often continuous with the endoplasmic reticulum and bear ribosomes as well. Nuclear envelope is interrupted at the places with minute pores. We call them nuclear pores. Why are these important? These nuclear pores help in the exchange of material. Which material? Like RNA, proteins, between the nucleoplasm and the cytoplasm. What is nucleoplasm? The part of the protoplasm found inside the nuclear membrane is called nuclear matrix or the nucleoplasm. It contains chromatin reticulum network and nucleolus. What is nucleolus? The contents of the nucleolus is continuous with the rest of the cytoplasm. It lacks the boundary membrane. They are the sites of active synthesis of ribosomal RNA, and they make ribosomes, right? Lack boundary membrane. This is about the nucleus. Now moving to the chromatin network. When we see chromatin network, chromatin contains DNA and the basic proteins called histones and non-histones along with RNA. Okay? When the cell starts division, this chromatin condenses into individual rod-like structures. We call them chromosomes. Every chromosome has a primary constriction known as centromere, which is a non-stained region, and holds the chromosomes together. Then we see kinetochores. These are the disc-shaped structures on the sides These are the kinetochores. We call them kinetochore. These are disc shaped on the sides of the centromere. Here the main purpose is the chromosome become attached to the spindle fibers by their kinetochores. Okay, and that helps in cell division. That means separation of chromosomes at the time of cell division, right? On the basis of the position. If the centromere, the primary constriction, is at the tip or at the notch, we call it telocentric. If the centromere shifts a little towards this side, one arm is short and another arm is long, we call this arrangement acrocentric. If still we have short and long arm arrangement but not exactly in the center, we call it submetacentric. And if the centromere shifts exactly in the center, and both the arms are equidistant, it bears a secondary constriction and a satellite onto the top as well. Here at this position we call it metacentric. Okay. Moving to the microbodies. Microbodies are spherical, oval vesicles with a single membrane boundary. Where do they occur? They occur both in plant and animal cells. Both in plant and animal cells. And what do they contain? Variety of enzymes. Why? Because these are responsible for carrying various metabolic pathways. Various examples are peroxisomes, these enzymes, glyoxysomes, and then on the basis of enzymes they are known as the different types of microbodies. This is all about this chapter.